Today I discuss about the DNA. DNA act as the genetic material. DNA act as the genetic material in organisms. Okay, it is a it is a nucleic acid. It is a nucleic acid. The full form of DNA is deoxyribonucleic acid. Deoxyribonucleic acid. So today I discuss about the structure of a DNA. Previously told that all nucleic acid are formed by many nucleotides. So the structural unit of DNA. So the structural unit of the DNA is nucleotide called deoxyribonucleotide. Four types of nucleotide are participate. Four types of nucleotide are participate to formation of DNA. These are the these are the DAMP means deoxyadenosine monophosphate. DGMP means deoxyguanosine monophosphate. DCMP means deoxycytidine monophosphate, and DTMP means deoxythymidine monophosphate. Okay, so these are the four deoxyribonucleotides which takes part to formation of a DNA. According to scientist Watson and Crick, according to scientist Watson and Crick, the DNA has the following characteristics. First one, it is the right-handed double helix comparable to a twisted letter. You all remember that in organisms DNA have many structures such as aDNA, bDNA, ddNA, zDNA, etc. Watson and Crick describe about the structure of bDNA in 1953. So, according to the Watson and Crick, they told that the bDNA is right-handed double helix comparable to a twisted ladder. It has two strands. Second one, it has two strands are composed of the deoxyribose sugar and phosphate only. So the strand of DNA are composed of deoxyribose sugar and phosphate only, which are attached through the phosphodiester bond. And the nitrogen bases are arranged inwardly. Okay. Then third, the two strands of DNA are anti-parallel. That is, one strand runs in 5' to 3' direction, while the another runs in 3' to 5' direction. Next, 4. The length of a helix is 34 Armstrong or 3.4 nanometer, and each helix contains 10 nucleotides. So the distance between two nucleotides is 3.4 Armstrong or 0.34 nanometer. Then 5. The diameter of a bDNA is 20 Armstrong or 2 nanometer. Then number 6. The number of purine base is similar to the number of pyrimidine base. That is, A plus G is equal to T plus C. Adenine plus guanine is equal to thymine plus cytosine. So, the number of A plus G is similar to number of T plus C. Here, a rules which is given by scientist Chargaff. According to Chargaff, the amount of purine base and pyrimidine base in a DNA is same. So we can express as A plus G is equal to T plus C is equal to 1. A plus G is equal to T plus C is equal to 1. So that is the expression of the Chargaff's rule. Then 7. The nitrogen bases which are arranged inwardly, the nitrogen bases which are arranged inwardly in between two DNA strands are attached by forming hydrogen bond between them. A double hydrogen bond is formed in between adenine and thymine. A double hydrogen bond is formed in between adenine and thymine, and a triple hydrogen bond is formed in between guanine and cytosine. So these are the characteristics of the bDNA according to scientist Watson and Crick. Here I drawn a structure, physical structure and chemical structure of DNA helix. So you look at the physical structure of the DNA, means that structure. Here two strands are present, and the strand are composed of sugar and phosphate by phosphodiester bond means two pentose sugar are connected with a single phosphate molecule by forming two ester bond. For that, this ester bond is called phosphodiester bond, because here two ester bond forms in between two pentose sugar with the phosphate. Okay, and look at here, the nitrogen bases such as A, T, G, C, Cg, Ta, Cg, Gc, At, Ta are arranged inwardly from the strand. Okay, then look, A and T are bound with double hydrogen bond. G and C are bonded by the triple hydrogen bond. Okay, DNA also has two groups. These are the it is called the minor group. It is called the minor group, and this is called major group. So DNA has two group. These are the minor group and major group. Okay, means the depth of the group is larger, then it is called major group. If it is smaller, then called minor group. A single helix means from here to here, a single helix. The distance or the length of a single helix is 34 Armstrong, and a complete helix is formed by or composed of 10 nucleotide. 10 nucleotide means 10 nitrogen bases are present in that place of DNA. Okay, so the distance between each nucleotide is 3.4 Armstrong, because the total length is 34 Armstrong and this helix contains only 10 nucleotide. For that, the distance in between two nucleotide is 3.4 Armstrong. Okay, and according to Watson and Crick, the diameter of the DNA is 20 Armstrong or 2 nanometer. Fine. So that is the physical structure. Look at that end. Look that at end. Here, a strand, that is strand runs from 3 prime to the 5 prime, and another strand is runs from 5 prime end to the 3 prime end. So that is the physical structure of a DNA. Then come to the chemical structure. Look at here. That is a pentose sugar. That is the 3 carbon place, and that is the 5 carbon place, and that is the first carbon place. In third carbon place and fifth carbon place and the third carbon place attach with the phosphate of two adjacent nucleotide. Okay, and the nitrogen bases are attached with the first carbon. Look at here. So that is the pentose sugar. Pentose sugar bind with phosphate in its 5' end. So the 3' end is free. For that, this end is called 3'. another another sugar means another nucleotide connected with the phosphate by its 3 carbon place. Then it attach with the phosphate by 5 carbon end or 5 carbon, and another means adjacent nucleotide is connected with that phosphate by its 3 carbon place. So this is a strand. So this is a strand which are composed of this is a strand which are composed of sugar phosphate only, pentose sugar and phosphate only, and the nitrogen bases, look at here, and the nitrogen bases are arranged inwardly. Another strand is runs antiparallelly, means 5' to 3'. One strand is 3' to 5', then another strand is 5' to 3'. And here the nitrogen bases are arranged inwardly and they are interconnected by forming of hydrogen bond. So that is the chemical structure of the DNA. Then look at that chart where I discuss about the different types of DNA, difference between the different type of DNA. Features: helix type. In ADNA, the double strand are right-handed. In BDNA also it is right-handed, but in ZDNA it is left-handed. Then diameter. ADNA has the diameter of 2.55 nanometer, BDNA has 2.37 nanometer, and ZDNA has 1.84 nanometer. Length of a single helix. In ADNA it is 3.2 nanometer, means 32 Armstrong. In BDNA 3.4 nanometer, means 34 Armstrong, and in ZDNA it is 4.5 nanometer, means 45 Armstrong. Number of nucleotide in a helix. Number of nucleotide in a single helix. In ADNA, the number of nucleotide in a helix is 11. In BDNA, it is 10, and in ZDNA, it is 12. So the distance in between two nucleotide in case of ADNA is 0.29 nanometer. In BDNA, it is 0.34 nanometer, and in ZDNA, it is 0.37 nanometer. So these are the difference between different type of DNA. Now some associated term. Now some term which are associated with the DNA. Template strand or sense strand. DNA has two strands, but all RNA are single stranded structure, and we all are knows that all RNA are produced from DNA. So the RNA the RNA produced from a single strand of a DNA. So the DNA strand which takes part in the RNA synthesis is called template strand or sense strand. Then coding strand or antisense strand. The another strand which does not take part. The another DNA strand which does not take part in RNA synthesis is called coding strand or antisense strand. Then denaturation and renaturation. Denaturation means separation of two DNA strand by degrading of hydrogen bond which are present in between the nitrogen bases. When does it occur? It occurs if we altering the pH of the medium or increasing the temperature of medium where the DNA is present. So DNA containing medium we can denature the DNA by altering the pH of the medium or increasing the temperature of the medium. The separation of two DNA strands by degrading of hydrogen bond called denaturation, and the aggregation of separated DNA strand is called renaturation. When renaturation will occur? If we decrease the temperature if we decrease the temperature or change the pH into the normal state, or we change the pH of the medium into the normal state, then the separated DNA strand gets attached. It is called renaturation. Then another term, it is melting point. It is melting point. Melting point means the temperature. Melting point means the temperature in which all DNA gets denatured is called melting point. So in which temperature the all DNA of a medium gets denatured is called the melting point. Now I discuss about the RNA. The full form of RNA is ribonucleic acid. Means it is composed of nucleotide which contains ribose sugar as their pentose sugar. So RNA is composed of so the structural unit of RNA are ribonucleotides. These are the 4 types, that is adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and uridine monophosphate. Previously I said that the RNA has 4 nitrogen bases. The nucleotides of the RNA contains 4 nitrogen bases. These are adenine, guanine, cytosine and uracil. Uracil is present only in ribonucleotide, means only in RNA. So these are the structural unit of the RNA. Then we discuss about the structure of different types of RNA. First I discuss about the mRNA. mRNA, M stand for messenger. So what is the function of mRNA? mRNA carries the message from DNA, means nucleus to the cytoplasm, and it also determines the arrangement of amino acid in a protein. Okay, so these are the function of mRNA. Now the structure of mRNA. mRNA is single stranded. All RNA are single stranded. So mRNA also the single stranded. So this is a mRNA strand. mRNA runs from 5 prime to 3 prime, 5 prime to 3 prime. At 5 prime end of the mRNA has a methyl group. The 5 prime end of the mRNA contains a methyl group. This region is called cap region. This region is called cap region. It contains a methyl group, means CH4 group. Okay. What is the function of cap region? Cap region attaches the mRNA with the ribosome at the time of protein synthesis. Okay, so it helps to bind with the ribosome. Then NCR, then NCR1 means non-coding region 1 is present just after the cap region. Then the initiation codon is present. Then the initiation codon is present, that is AU and G. AUG. This portion is called initiator or initiation part of the DNA. Means from that codon, what is codon? Codon is the arrangement of 3 nucleotide, arrangement of the 3 nitrogen bases, arrangement of the 3 nitrogen bases on a mRNA is called codon. Okay, so that is the initiation codon or start codon, which is present just after the non-coding region 1. Then the mRNA contains a coding region, so that parts only of the RNA of the mRNA participate in protein synthesis from AUG to the end of coding region. So this portion, this portion is called coding region. Then the stop codon is present. Then stop codon is present. Stop codon is present. Here 3 codon will present as stop codon. These are the UAA, UAG and UGA. UAA, UAG, UGA. From these codons, only one is present at that site, means at the site of the stop codon. It stops the protein synthesis. It stops the protein synthesis process. So protein synthesis start at that point and ends at that point, because stop codon does not codes for any amino acid. For that amino acid, means for that the formation of peptide chain is end or will end at that point. So that is the functional part of the mRNA which takes part in protein synthesis, but these are also useful part. Without this part, the protein synthesis does not occur from the mRNA. So then a then the next part of the stop codon is NCR2, means non-coding region 2. What is the function of non-coding region 2? If the protein synthesis does not stop, at the stop codon, then the ribosome also reads the codon. For that, protein synthesis does not stop at that point, and they move forward, and they move forward. If they move forward, then the second region means NCR2 prevent the attachment of amino acid to make a peptide bond, because the codon of that part, or the nitrogen bases which are present in the non-coding region 2 does not code any amino acid. For that, protein synthesis, if does not stop at that point, then it must be stopped at that point. Okay, then at the end of 3' of the mRNA, it contains many adenine nitrogen bases, and this portion is called poly poly A tail. Why it called poly A tail? Because many adenine or adenine nitrogen bases are present repetitively at that portion. Okay, so that is the structure of mRNA. Then I discuss about the RNA, rRNA. What is rRNA? R stand for ribosome. So the full form of RNA is ribosomal RNA. Okay, you all knows that the rRNA synthesized by the nucleolus. For that, the site of formation of ribosome is nucleus. Okay, then the structure of rRNA also a single stranded structure. rRNA also a single stranded structure, like that. Look, it also runs from 5' end to 3' end. 5' end to 3' end. You look here, a loop is formed in rRNA. Why? Because the RNA contains the complementary base sequences. rRNA contains the complementary nitrogen bases. For that, sometimes they form the loops by forming hydrogen bond in between the nitrogen bases. But it does not the double stranded. Always the RNA is single stranded. Okay, so that is the structure of RNA. Different types of RNA takes part. Different type of RNA takes part in formation of ribosome. You all knows that the ribosome has two part. That is smaller subunit and large subunit. We also discuss about the ribosome in the cell chapter. Okay, in the cell chapter. Again, I repeat it. The ribosome has two part. Means a smaller subunit and a large subunit. Okay, small subunit and a large subunit. Ribosome also classified into two groups. These are the prokaryotic ribosome, means 70S ribosome, and the eukaryotic ribosome, means 80S ribosome. The 70S ribosome has a smaller subunit and a large subunit. The small subunit of the prokaryotic ribosome is 30S, and the large subunit is 50 S. And in 80S ribosome means eukaryotic ribosome, it has the 40S as small subunit and 60S as large subunit. Then 30th subunit of 70S ribosome is composed of 23S rRNA. 23S rRNA. Sorry. 30S subunit is composed of 16S RNA. It's composed of 16S RNA. And the 50S ribosomal subunit means large subunit is composed of 23S rRNA and 5S rRNA. Okay. In 80S ribosome means prokaryotic ribosome, the smaller subunit means 40S ribosomal subunit is composed of 18S rRNA and the 60S subunit is composed of 28S rRNA, 28S rRNA, 5S rRNA, 5S rRNA and 5.8S rRNA. 5.8S rRNA. Look here. In both the prokaryotic ribosome and eukaryotic ribosome has 5S rRNA. It is the common part. 5S rRNA is the common part between prokaryotic ribosome and eukaryotic ribosome. Why the common part is present in both? Because at the time of protein synthesis, ribosome gets bind with the cap region of the mRNA, and at that bonding and in that bonding, the 5S rRNA takes part. 5S rRNA takes part in attachment of ribosome with the mRNA of the cap region of the mRNA. For that, both prokaryotic and eukaryotic ribosome contains the common part 5S rRNA. Now I discuss about the tRNA. T stand for transfer. The full form of tRNA is transfer RNA. So the tRNA transfers the amino acid from the amino acid pool of cell to the site of protein synthesis. Means, look, if it is a cell, then amino acid present here, it distributed all over the cell. That is the nucleus of the cell. Amino acid present or distributed all over the cell cytoplasm, and protein synthesis occurs at that point, that is the mRNA and that is the ribosomes. Then the protein will formed by the formation of polypeptide chain, and the polypeptide chain is also formed by the polymerization of the amino acid. So amino acid are needed at that site where protein synthesis will occur, and tRNA carries the amino acid to that site from the amino acid pool of the cell. Okay, so the RNA which carries the amino acid from amino acid pool of the cell to the site of protein synthesis is called tRNA or transfer RNA. Okay, then the structure of tRNA. As same, tRNA is a single stranded structure which runs from 5' end to 3' end. But this RNA, this RNA has many complementary nitrogen bases. For that, they folded. They folded a proper manner. Like that. Okay. So 5' and 3' comes closure. Okay, and the strand has many complementary bases. For that, they forms 5 arms in the tRNA. If we enlarge that structure, then and this structure of tRNA described by scientist R.H. Hole, and this model is also known as clover leaf model. This model is also called clover leaf model, proposed by scientist R.H. Hole. If we enlarge that structure, then we can observe like this: the tRNA has like that. So that is the 5' end and that is the 3' end. The 3' end of the tRNA contains a group C C A, and the adenine has a free OH group, At that point, the amino acid get attached with the 3' end of the tRNA. For that, this arm is called this arm of the tRNA is called acceptor arm, because this arm accept the amino acid. Or this 3' end attach with the amino acid. Okay, for that this arm is called acceptor arm. Then the second arm has formed a loop. Why does the loop will form? Because at that point has no complementary. That part of the tRNA, that part and that part do not contains any complementary nitrogen bases. For that, this does not form or cannot form the hydrogen bond. For that, this part of the tRNA formed a loop. This arm is called T-psi loop. Okay, because this arm contains pseudo uracil group, unusual base called pseudo uracil. For that, this loop is called T-Shice loop. Then a variable arm is present. A variable arm is present in tRNA. Why it is variable arm? Because the length of that arm, does not fixed. Okay, it is called variable arm, and it's composed of 7 to 11 BP. It contains 7 to 11 nitrogen bases. Okay, BP means base pair, because here the nitrogen bases present in pair and they form the hydrogen bond. For that, the length of the variable arm in between 7 to 11 base pair, and it varies in RNA, in different RNA. For that, this arm is called variable arm. This arm has no important role in protein synthesis. Okay, that is only formed for the complementary nitrogen bases. And the next loop is called anticodon loop or anticodon arm. This loop is called anticodon loop or anticodon arm. Why? Because it contains 3 free nitrogen bases, means codon, such as the initiation codon of mRNA is AUG. So here U A C, which are complementary to the AUG bases. So this codon is called anticodon. And for the presence of anticodon, this arm is called anticodon arm or anticodon loop. Then the last term of the tRNA is called DHU loop. This term is called DHU loop for the presence of a unusual base named as dihydrouridine. Okay, so in tRNA we can see 2 unusual base naming as pseudouracil and dihydrouridine. For the presence of dihydrouridine, this loop is called DHU loop. Then come to the function of different arm or different loop of the tRNA. First, acceptor arm. Acceptor arm binds with amino acid. Okay, for that the tRNA carries the amino acid. If the amino acid does not attach with the tRNA, then how does it possible for carrying the DNA? Acceptor arm binds with the amino acid. Then the function of T-Shice loop. T-Shice loop helps in attach with ribosome. T-Shice loop. Okay. Then the anticodon loop. Anticodon loop has 2 roles. First one is it binds with the mRNA. And another is it determines the amino acid. means the amino acid which is attached which will attach with the 3' end of the tRNA is determined by the anticodon or anticodon arm. For that, the function of anticodon loop is binds with mRNA at the time of protein synthesis, and it determines the amino acid. Okay, and it determines the amino acid means which amino acid gets attached with which tRNA. It determined by the anticodon arm or anticodon. Then DHU loop. DHU loop also has an important role because it synthesizes the amino acyl tRNA synthase enzyme. It synthesizes the enzyme named amino acyl tRNA synthase enzyme synthesized by the DHU loop. What is the function of this enzyme? This enzyme helps to bind the amino acid with the tRNA. Without this enzyme, an amino acid does not attach with the tRNA. Okay, so that is the function of DHU loop. Then hnRNA. hnRNA. Full form of hnRNA is heterogeneous nuclear RNA. Heterogeneous nuclear RNA, which is the intermediate RNA between DNA and mRNA. So if we imagine this is a DNA strand, this is a DNA strand, this is the DNA strand, and RNA synthesized from the template strand of the DNA. Previously I told that the RNA synthesized from the DNA or produced from the DNA. So from DNA, a single-stranded RNA will produced. This RNA is called hnRNA. Why? Because after the transcription means the process of forming RNA from DNA. The process of forming of RNA from DNA is called transcription. After transcription from DNA, a hnRNA hnRNA will form, which has two parts. That is a exon part and an intron part. An exon part and an intron part. Then another exon part, another intron part is present in this RNA. So the sky color part is exon and this part is intron. intron. Exon is the functional part of mRNA, of RNA. Exon is the functional part of RNA and intron is the inactive part. Exon is the active part of the RNA and intron is the inactive part of RNA. So at the time of mRNA formation, the a processing will occur. A processing will occur. This processing is called this processing or this process is called splicing. Splicing. Splicing is occurred by help of many enzymes such as ligase and nuclease. Ligase and nuclease. Nuclease cuts the intron part, mean inactive part from the DNA. Cuts the nuclease enzyme cuts the intron part from the RNA. And ligase bind the all exon part of hnRNA, and a active or functional mRNA will form. So what happen at that state? All intron part are get separated by help of nuclease enzyme. By help of nuclease enzyme from the hnRNA, that is the exon part. These are the exon part. When the intron part gets separated from the hnRNA, then ligase enzyme attaches the exon part only and forms a functional mRNA. So it is the functional functional mRNA. Then what is hnRNA? hnRNA is the intermediate RNA which are produced at the time of transcription, means at the time of formation of functional mRNA. Okay. Thank you.